Welcome to the Secular Hub. We're so glad you came out tonight. And I know that many of you are here, or some of you are here for the first time, so let me say a couple of words about the Secular Hub. We are one of the few in the country secular organizations that has an actual meeting space. So we have all kinds of programs. If you look on our website, you will see that we have book clubs, game night, movie night, coffee, Sunday coffee, all kinds of things. So we hope to see you again at one of our events. The Hub was officially opened 7 years ago on Darwin Day in February of 2013. So we're going to have our birthday next month. We're going to celebrate Darwin's birthday and the Hub's 7th birthday. Have a cake, we have a liquor license, we'll have beer and wine for sale, so we hope you join us then too. And we're funded strictly by membership dues, well, mainly membership dues. So if you aren't a member and if you would like to drop a little something in that little box on your way out, we would greatly appreciate it. I'm Evelyn, by the way. I chair the events committee. And we put on a science program like this one every second Saturday from 5 to 7. And the man that's in charge of that, we're very fortunate to have somebody of his caliber do it, is Clyde Zadens. Until a couple of weeks ago, he chaired the physics department at CU Denver. He's a graduate of Caltech, a professor emeritus, and Clyde will now introduce our program.
Thank you, Ev. One just quick other announcement. In March, we're going to be doing a talk on the mathematics of music. And of course, next month, with the Darwin Day birthday, it's going to be Lee Shapiro talking about bringing Darwin into the 21st century. So tonight's speakers are members of a group called the Project Bridge. And Dylan Verdon is here, and he's going to say a little bit about that group of people.
Hello all. Thank you for coming here. It's nice to see such a full crowd. Today's event is actually going to be from the conjunction of two groups. The first, as Clyde mentioned, is Project Bridge, which is a student-led organization that really focuses on training early career scientists to engage with their community, and that may be through policy or through community communication events. And we really try to focus on making sure that the scientists that we train are able to actually interact with the community in a meaningful way. And I'd like to thank you for being the second half of that and really helping us out. The second group is called the Neuroscience Outreach Group, or NOG, and it's kind of an organizational name that's been given to some long-standing efforts by our neuroscience students to reach out to the community and tell them how the brain works, what do we study, what is the life of a scientist like. One event that you might associate with that is during the free museum day at the Denver Nature Museum Science. Obviously, I'm not the speaker tonight. We have a free science day where we have exhibits that teach young children about how the brain works through different sensory systems and different ways of learning. And so between these two groups, we're now kind of trying to get more neuroscientists out into the community and to talk to you about the brain. And so we're going to go through a couple of different talks over the course of the day. And Clyde, were you going to introduce those?
Well, thank you, Dylan. The topics tonight are neurons, Glia, and the gut-brain axis. Now, this is unusual. We normally have one speaker, so there are three speakers tonight. So this is a brand new way for me to try to introduce people. So the first talk is going to be on the neurons, and Dr. Courtney Wilson is going to be the speaker for that. I think what I'll do is I'm going to introduce the three people, and then hopefully we'll have a seamless transition when one talk ends and goes to the next one. So Dr. Wilson is a taste neuroscientist at the Anschutz Medical Campus. She received her PhD in 2018 and continues to be passionate about both brains and science communication. The second speaker is Dr. Caleb Dahl, if you want to raise your hand or something. He's a native Kansan with a long-standing interest in scientific research. He received his PhD at Vanderbilt University in Nashville. Currently, he's a research assistant professor at the Anschutz Medical Campus. His research centers on genetics underlying myelin sheath growth. Outside of the laboratory, he enjoys music, literature, and chasing his 2-year-old son around the city. He's going to be speaking on the Glia part of the talk. Our third speaker is Sarah Cook. Sarah is a 5th-year student in pharmacology PhD program at CU Anschutz Medical Campus. She studies the underlying basis of learning and memory at the cellular level, with a particular emphasis on Alzheimer's disease. In her limited free time, she enjoys reading and long hikes with her dog. So we'll start out with Dr. Wilson on neurons.
Great. I already have this,
Oh, you did it.
so I don't even need that. Awesome. Thank you. Can everyone hear me okay? All right, great. And if you can't, just like holler at me and I'll fix something. So like was said, I'm Courtney. And I'm here to talk to you about neurons. Just a little bit about me. I did my undergrad really close to home in Boulder, majored in molecular biology, among other things, and took this course my senior year that was all about how brains work and the molecular basis upon which they work. Ooh, exciting. And found that that's where all the action was, because that's how everything works. How we understand the world, how we process sensory systems, et cetera. So I really fell in love with the brain and decided to go to grad school for that. So I went to grad school out here at CU Anschutz, where I graduated and currently am working as a postdoctoral fellow in a lab that actually works on taste, as Clyde mentioned. So I'm trying to figure out how taste buds work. But today, I'm going to focus on the neuron. These are actually all neurons that you see here. It's not a Klimt piece of artwork, although it kind of looks like it. So what happens, this is actually an image from an actual mouse brain. And mice don't usually have nice, pretty rainbow neurons, but this particular mouse has been genetically engineered to have its neurons light up in different fluorescent ways. So we get this really nice image here. And I hope to convince you that neurons are awesome and totally worth studying. So let's start. Your brain is a very busy thing. Everything that you do, whether it's listening to your favorite piece of music or any other sensory experience that you're having, listening to someone drone on about neurons for a while, or thinking, processing emotions or memories, or, thinking about democracy or something highfalutin like that, or whether it's moving. So all of the actions that your body takes in the world, they're all of these things are rooted in your brain's activity. So it's a pretty special organ. I may be biased, but it is. And the basic job of the brain is to take sensory information from the environment through, sight, hearing, touch, etc., process that information along with memories and emotions and, markers of internal state, if you're hungry, say, or not hungry, and use those things to make decisions and eventually implement actions in the world to move your body so that you're interacting with your environment in an appropriate way and, hopefully continuing to survive on the planet. Which is, all of our goals, I feel like. So brains, we're going to talk to you today about 2 different types of brain cells. They're the 2 main cell types in the brain. I'm going to tell you about neurons, which are the star of this particular talk. And they're responsible for getting information around your brain all the time. And then Caleb's going to tell you about glia, which are oft neglected and not talked about very often, but are super, super important for, one, the function of neurons, but also just generally your brain health. So we're going to cover those today. So what is this cell? This is a weird looking cell, right? It's not your typical, egg shape that you got in biology in high school that has all the little bean mitochondria and that stuff. It looks pretty weird. So these little bits up here that look kind of like a tree or fingers are called dendrites. And their job is to gather information from, whatever comes before that particular neuron and process that along with the cell body. So every cell has to have a nucleus, right, where it keeps all its DNA. It needs organelles to keep functioning. A lot of that stuff goes in the cell body. It's kind of the chunky part of the neuron. And then we have this really long bit of the neuron called the axon. So that you can sort of think of as the wire of the cell. So it's taking information from what I would call these processing parts up here and sending it on down to the other end of the cell. So you folks have probably heard of gray and white matter, I assume. Raise your hand if you have. Good, good. You're awake. So what we traditionally think of as gray matter is actually the muck of the cell body and the dendrites, the kind of chunky stuff of the cell, whereas the white matter is this axonal track here. So that's the wire of the cell, is the white matter. So if we look at this picture of a brain, this is a picture through a human brain, so cut about here. And what you can see are there are these slight differences in the shading of the tissue sort of naturally. So all along the outside here of this section, you see darker material than the inside. So this outside material is the gray matter, and that has a lot of cell bodies and dendrites in it. It's where a lot of the processing goes on. And this white matter on the inside is actually made up almost totally of axonal tracks. So it's essentially the highways of information between parts of your brain, eventually out to the rest of your body. And that's how the whole thing is sort of structurally organized to get you oriented. Now, what's the point of this cell? What is it doing? As I alluded to with some of the functions of these parts, the point of a neuron is to get information from one end of the cell to the other end of the cell, right? And that information can change depending on what part of the brain it's working in, whether it's a sensory sort of neuron or a processing sort of neuron or a motor neuron, etc. But the point is to get information from one end, where the information is gathered by the dendrites through the cell body and out the axon. Now, this is accomplished using electricity. So just like a sort of electrical wire system works, electrical impulses travel from one end of the cell to the other. That's how your brain works. But when it gets to the end of that cell, if it tries to talk to another neuron, the electricity can't actually transfer because they don't share a membrane, and that's how the electricity transfers along the cell in the first place. So at that point, it's got to become a chemical signal. So these chemicals are called neurotransmitters. I'm sure you've heard of a lot of them before. Serotonin, dopamine, GABA, glutamate, all those words refer to different neurotransmitters, which make up those chemicals that are transferred here between these two cells. And that way, this cell can gather that information with its dendrites and make that signal once again into an electrical one. So this section right here, where the signal becomes chemical and not electrical, is actually called the synapse. So whenever you hear synapse, that's what it's talking about, two neurons contacting one another and communicating via chemicals. So neurons are super exciting. One of the great things about neurons is that they are incredibly diverse. So the one you usually see, I'll go back for a second, the one you usually see has this cell body, has some dendrites, has a really long axon. This is a typical structure for a motor neuron. But your central nervous system is full of all kinds of neurons. And they look pretty different from each other, and that depends on the function of those particular cells. So some, like this is a Purkinje cell, which is in your cerebellum. It's kind of like the little backpack on the back of your brain. And it processes a lot of sensory and motor information to better estimate where your body's supposed to. It programs fine movements, basically. So it needs a lot of this processing power to take in a lot of different sensory information from different parts of your brain. This one is smaller and actually is an interneuron, so it communicates between two neurons in a circuit in your cortex. This one is also one that's in your cortex. It's what we call a pyramidal neuron. Does also a lot of processing. So basically, all of these different structures are so diverse because the cells need to be so diverse. Some are in your eyeball, and they need to have this flat bit here that's registering a bunch of information from rods and cones of your retinas, and before it sends it on down to the back of your brain where vision is processed. So form really follows function here. So they're all really different in size and length also. So a lot of these aren't pictured, but neurons can be actually quite long. You think of cells as super small, but there's a neuron that goes from the base of your spinal cord all the way down to the tip of your toe. So that's one cell, which is pretty impressive. I know. And there are about 80 billion of these, and they are all different from each other. So there's a lot to work with and a lot to study here for neuroscientists. Will be employed for a while, hopefully, if we all keep funding the NIH, which we should do. But so neurons are diverse in shape. They're also diverse in the kind of rules they play by. So some neurons need one input. So one neuron fires, the next neuron fires. We use the word fire to denote activity of a neuron. And they don't need that much electrical input to continue on the message. But some need more. So they need like 2 neurons to say, hey, there's information here at the same time for that neuron to then continue on the message through the circuit. So these are really complicated and, importantly, really diverse. And not only are they diverse, they are also dynamic, which is super exciting. So this is actual real data. Ooh, exciting. Oh, I sent the earlier one. Anyways, so this is a part of a neuron. It's a dendrite that a classmate of mine before we both graduated worked on, and he focused, like Sarah does, on memory and learning. So what you'll see in this video when I play it in a second is there are these, along this tract, there's a lot of these little nubbin things. So those nubbins are where the neurons actually form synapses with other neurons. So they are the points of contact. And what's going to happen in this video, it's going to be hard to see. Maybe I can move it. There's a little blip of light, which denotes when he is actually stimulating the neuron. And you'll see that these little nubbins actually kind of get brighter. And that's because the neuron is physically changing in response to activity, which is what our brains are doing all the time when you're learning. So I'll go see if I can play this video. Boop, 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 boop. So yeah, I'll let you know when the blip comes. So focus on those little nubbins. Whoop. You want to play? Oh, it says the QuickTime is not available. Bummer. It's not a big deal. You can just imagine them getting bigger. I promise I'm not lying to you. It's true. Alas, videos and PowerPoint, you know, always and forever a weird combo. But yeah, so basically, we have this saying in neuroscience that's neurons that fire together wire together. So if you have a really repeated, like if you're remembering something, if you have a really repeated contact between 2 neurons, the point of that contact, the synapse, will actually grow bigger and change in its composition so that that communication is faster and more efficient. So when you're doing something like practicing piano, you're actually going over those circuits over and over again in your head, and that's why you get better at piano, which is pretty cool. So they are diverse, they are dynamic, and they are also make up really, really complex circuits. So the next slide is scary, and I'm just warning you now. There we go. So this is your auditory system, and you are not supposed to be gleaning anything super exciting from this besides that this is really super complex, right? So all of these little boxes pertain to different parts of the brain that are involved in auditory processing. So every time you're hearing something, this part's activated, this part's activated, this part's activated, and they're all doing different things in this really complex circuit. So what neuroscientists are trying to do every day is figure out how the heck these are connected, what are the rules upon which they're connected, what changes about them, and how they form. So figuring out these really, really complex structures is what we do all the time. So to go through a sort of more simple structure, I'm going to talk to you about a little bit about sound localization. So sound localization in a barn owl, or indeed any birds, works by a pretty simple circuit, which is fun to go through. So sound localization works in part because of timing differences of sound. So if I'm standing here and there's a sound coming from, say, the kitchen, it'll hit my right ear a little bit before it'll hit my left ear, just based on the fact that I have a head and 2 ears. And so my brain can use that difference in timing to figure out where the sound is, right? So this circuit looks a little bit like this. A guy came up with it actually before we figured out that it was real in birds. It's different in mammals. Sorry, your brains don't work like this, just his does. So what happens in this circuit is we have a bunch of neurons here, A, B, C, D, and E, all represent neurons. And the rules of these neurons is that they don't pass on any information unless they get 2 synchronous inputs. So they got to be stimulated by 2 different inputs. And the things that stimulate them are, there you go, are the different inputs from the ears. So we got the right ear over here, the left ear over here. And these neurons play by the same rules as each other. So they travel, they do the same speed, the same everything, basically. So if there's something directly in my center and I were a bird, which I'm not, but I am for this thing. I look like that. Pretend I'm like that. And there's something directly in front of me and making a sound. That sound is going to hit both of my ears at exactly the same time, right? So if you kind of imagine the signal hits here at the same time, goes there, and they coincidentally hit this neuron C. So neuron C then would sort of encode and let the bird know, hey, something's directly in your center. Whereas if something's, say, much closer to my right ear, it'll hit this ear first. So it'll get to go here, before this gets, and then it'll coincide on a different neuron. Does that make sense? Kind of, sort of. I know it's a little bit complex. Yeah, so it's a pretty cool, almost very simple circuit to figure out where something is in space. So if, the center neuron gets activated, it means something's in the center. This means it's to the right. This means it's more to the right, etc. So that is how the bird circuit works. And this took years and years and years for neuroscientists to figure out that this was actually real. And so this is the kind of work that we're doing all the time to learn about this. And so since we have all these complex circuits and that underlies everything we do, you can imagine that things go wrong sometimes, right? So what happens when these neural circuits don't function properly? So let's take stroke, for example. Whether you're having a stroke that is depriving your neurons of oxygen, which they need to operate, or flooding them with blood, which they also don't like, those circuits are going to be disrupted because the neurons can't function and start to die, right? So what happens in where you have that either lack of blood or too much blood, in that region, the circuits that are located there will be disrupted. So if someone has a stroke, say, in Broca's area, which is where language is processed, and it's a pretty common place for people to have stroke symptoms, they will lose the ability to create language, not to understand it, just to form it, because the circuits that underlie forming language are actually being pretty heavily disrupted in this situation. Same thing is true for Parkinson's disease, right? So we're looking at a brain from the bottom. This is where the brainstem comes out later, and the cerebellum would be here. And these are actually your olfactory bulbs. That's a fun fact. But deep in the middle of your brain, there are these midbrain regions that control movement. So there's this one called the substantia nigra, and it usually has this nice little row of neurons that are busy making dopamine, which is one of the neurotransmitters that I mentioned earlier. And it's super important for movement in this particular circuit. So when in Parkinson's those neurons are actually dying off, and there's not enough dopamine, and the circuits are disrupted. So since this part of your brain controls movement, it results in the tremors that we see in patients of Parkinson's disease. So this is what happens when sometimes function gets lost, right? But it can go the other way also. So how many of you have heard the idea that we only use 10% of our brain? It's pretty pervasive, right? It's poppycock. So that's a scientific term. Don't believe it. So it's really, that idea has been propagated throughout like movies like Lucy and Limitless, where people are using the untapped 90% of their brain. That's bad. Don't do that. Because you don't want all of your brain active at the same time, right? Like all of your brain does get activated, but at different times. When you're playing piano, you don't want your soccer leg movement neurons to also be firing, because that'll be a mess, right? And we do actually see when neurons get overactivated too much in either a region or all of the brain, and it's called a seizure. So really don't buy this 10% crap, because it's not real. So obviously you don't want full synchronous activity of your whole brain, because that, as I'm sure you know, because of epilepsy, has a lot of symptoms of its own. But we like it in sort of the happy medium of every neuron has their happy place of how active they are and in response to what, and et cetera. So yeah, I hope I've convinced you that these little neurons are really exciting to study and super fun and complicated and worth our time and thought. And with that, I guess I'll take some questions and hand it off to Caleb. Yeah.
Well, I was thinking it might be a good idea.
Oh yeah, do you want to?
Questions at the end.
Cool, we can do that too.
I think that probably would be a smoother way to go. So just remember your questions.
Good luck.
Don't forget half my question. The memory one will be
The memory one will be good. Right. Exactly.
Thank you, Courtney.
Yeah, absolutely. Thank you guys. Oh yeah, I meant to say at the outset, thank you so much for having us here. We love talking about science, so it's fun to, do something other than just annoy our friends with random facts. So, thank you. It's on stick. You got this.
Higher? Okay. Just do that.
All right. Thanks again for having us. I'm a developmental neurobiologist, so I've always been interested in how the brain forms, how neural circuits form, how all these different cell types come to be, and how these cell types interact with one another. So we're going to talk about that a lot today. And I just have a few take-home notes here on the front slide here, and I will get this video going because it's pretty cool.
Wait, it doesn't need hooked up.
I don't think it does. So these are glial cells, and there's a particular type of glial cell called oligodendrocytes that I'm going to talk about a lot today. And this is the live video from a zebrafish spinal cords. So this is what we study in lab. And you can see how these oligodendrocytes emerge from this ventral part of the spinal cord, so the base of the spinal cord, and then they move dorsally up, and then they're eventually going to make connections with specific subsets of axons. Get that going again here. Well, okay. So this is one aspect of what we study, and really what I want to hit home today is, again, how these different cell types are going to interact with one another. So we can run through that one more time. So I've just kind of pseudo-colored a couple of these cells as they emerge out of the spinal cord there. These embryos are about 4 days post-fertilization, so we can study zebrafish embryos just in a brief time period of development for them, but this would take us many, many months in a human context. Okay, let's get rolling here. So just like Courtney said, there's a lot of different types of neurons. We also have a lot of different types of glia. And basically, the take home here is that any cell in your brain or in your central nervous system that's not a neuron, we can consider a glia. And we're really going to hit home today this aspect of this association with other cell types. So at their basic level, they are trying to help out our neurons. And I'm going to tell you a couple of different ways on a couple of different cell types on how they both support the function of neurons. We also have these really cool clues that they're actually helping support the formation of these connections, these synapses that Courtney set up for us. And they're also hallmarks of disease when neurological disease sets in. So we're going to talk a whole lot about these oligodendrocytes and a little bit about these really cool cells called microglia as we go. Okay, so we've got our different types of glia on this slide. Again, we're going to talk mostly about oligodendrocytes. That's one of these types of macroglia. These oligodendrocytes do a really fascinating thing where they wrap and insulate those axons that Courtney set up for us. So this insulation is really crucial for the propagation, the rapid firing of those action potentials. And then we'll also talk a little bit about these microglia, which are actually cells that come in outside of the central nervous system. They're formed and specified outside the CNS, and then they come in and they're the kind of immune cells of the central nervous system. So I'm going to pitch them kind of like as a Pac-Man. They're really necessary to go in and eat up any dead or dying cells or debris or those kind of things, and they're crucial for both the formation and the function of the nervous system as well. Okay, so this is broadly speaking. We've got some other cell types that we're not going to mention today. Astrocytes are also really fascinating and involved, again, with formation and function as well. Okay. So first we're going to talk about these oligodendrocytes. These are near and dear to my heart. They're illustrated here in green. We've got our neurons. As Courtney set up really beautifully, we've got these dendrites where they're gathering their information, and then they've got to send that information all the way down to the end of their axon. And sometimes, as she said, these are really, really long axons. So one way to make that highway more efficient and faster, the communication improved, is by wrapping those wires or those axons in insulation. So this is simply just a lipid-based wrap that goes around the outside of the axon, and it helps those axons fire. So the most hallmark disease that you all will probably be familiar with is if we start to lose myelin, that's the hallmark of multiple sclerosis. So that's the absence of these myelin sheaths. Initially, they have just a hard time with the propagation, but it can also lead to the death of those neurons as well. So that's kind of the most common disease that oligodendrocytes are associated with. Okay, now this is a cool slide to help illustrate another cool feature of what myelin allows us to do as vertebrates. So there's a lot of cool literature on these giant squid, because early on neuroscientists were trying to figure out what's the code between how these neurons know how to fire. Well, they found this giant squid that had one big axon, and this is just a cross section down through the length of the organism here, and you can see that it's got this one central huge axon. And it evolved that huge axon so that it can fire really quick action potentials. And now, while that's really great, giant squid, more complex organisms such as humans have to pack a whole lot more information into our nervous systems. So this would be a cross-section of the human spinal cord, and just like Courtney set up earlier, we've got both white matter and gray matter. And so these cells are on, well, actually the cells here are on the inside. These are all the different neurons, and all of this white part on the outside are these tracks. These are myelinated tracks. Not all of them are myelinated, but these are highways that are just shooting up and down your spinal cord. There are millions and billions of neurons here. And so if we can myelinate those axons, we can actually, they don't have to grow into a huge diameter of an axon. We can pack in a whole lot more information and a lot more cells by myelinating these neurons. So this is another awesome feature of vertebrate evolution. Okay, so I think we pretty well set up what myelin is, and let's go back to talking about neural circuits. And this is just going to be a really simple neural circuit, and we'll keep with this kind of sensory idea that Courtney was talking about as well. And again, she mentioned that neurons that fire together wire together. So let's say we've got these sensory neurons, A up here and B down here, and then we're going to have a motor output. And since I study zebrafish, and this is a good way to talk about them, so zebrafish, like all fish, I think, are really skittish. So it doesn't take a whole lot of sensory input, whether it's a loud bang or some disturbance in the water, but they're going to want to escape. They're always skittish. But for our example here, we've got these two sensory input neurons, and you'll notice that number A is actually at a farther distance away than sensory neuron B. Now, if these two sensory inputs have to come together at the exact same time, well, that means that this impulse from A has to travel a lot farther to get to that synchronous output. Well, one of the cool things that we've learned about oligodendrocytes is that they can come in and help speed up that connection by wrapping this neuron. They can make sensory input A fire more fast, and then they can have a synchronous arrival. So this implicates oligodendrocytes in a process called synaptic plasticity. So an outside cell, these glial cells that everybody thought were just these kind of dumb support cells, are actively involved in plasticity. They're facilitating the formation of this neural circuit. So this is a really cool advance to show that it's not just neurons doing all this business by themselves. We've got these support cells to help facilitate that development. Okay, so let's get back to our zebrafish model. This will set up a couple more things we're going to talk about today. So you can see the whole zebrafish here, or most of it. Its head is up here. It's got its eye here, the really complex part here, the brain and the hindbrain. And then what we study down here in the spinal cord, largely because these circuits are a lot more simple and we can look at individual cells. So this is a transgenic zebrafish, meaning that it's got transgenes integrated into its genome so that we always see these cells. We can also do techniques to just look at one individual oligodendrocyte or a neuron or whatever, just one at a time. And this allows us to kind of assess what kind of perturbations, what kind of genes are required for this oligodendrocyte to make a normal myelin sheath. Okay, so with this, we can attack different conditions. So one of the things that's really starting to pop out in the literature that a lot of different neurodevelopmental or neuropsychiatric diseases are now being associated with alterations in myelin. So there's a big list here on the side. I'm going to talk here very briefly about some modeling what we're doing on the autism spectrum. So we have a single gene disorder called Fragile X syndrome, and we can actually make Fragile X zebrafish because their genes are very well conserved all the way up to humans. And we've found that when we look at individual oligodendrocytes, those guys that are lacking this gene have very short myelin sheaths. So this would be referred to as hypomyelination. They're not making enough myelin. And a lot of you may be familiar that autistic individuals have a lot of trouble with these sensory domains. So they have trouble with bright lights or loud sounds or those kind of things. So hypersensitivity to these sensory domains. So we're now wondering if these deficits in myelin production are also linked to those kind of deficits in the sensory processing. Okay, so that's what one of the things we're working on in the lab. And next we're going to, I did want to mention as well that this isn't just a developmental process. So I am a developmental biologist. We're looking really early in development. But we actually continue to make myelin our entire lives. So they found that when individuals learn new skills, like learn how to juggle or play a new instrument, they're actually making new myelin in their brain. So this myelin is necessary for as we continue to learn and grow throughout our lifetimes. So it's not just something that happens early in development. Okay, and now I'm going to add a little bit more complexity because I want to add in this flavor of these microglia. And I'm going to talk about these microglia as actually helping the oligodendrocytes. And so I've talked about everything supporting these neurons, but I'm saying that these associations are so layered and these glial cells are so important on so many different levels. So broadly speaking, we can think about these microglia as Pac-Man in our central nervous system. So any kind of dying cells or debris, leftover neurotransmitters, they get consumed by these microglia. And they're always surveilling their atmosphere. So this is some pictures that were taken by a graduate student in our lab who's very talented. And she looked at these microglia. So those are labeled here in our yellow. And then those myelin sheaths that I just introduced to you, they were mostly in green in those previous slides, are all labeled in red here in the back. And you can see how this microglia just kind of lays over these myelin sheaths. And just kind of, she's taken videos of this as well, to see that they are just constantly looking for any kind of perturbations or some kind of signal for it to consume this myelin. So this is going to be illustrated here in this next slide. So she took this time-lapse video, and I'll set this up for you again. So this is a microglia, again in yellow. Our myelin is labeled here in red. And so you can see this myelin sheath that starts out here on the left part of this time series. And you can see by the end that it's been consumed by this microglia. So these glia are not, we've also known for quite a while that it's helping shape those synapses and the neuron connections as well. But she's now got good evidence that they're actually shaping the myelin as well. So this adds another layer of glia helping themselves and helping out one another to help shape nervous system development. So the really dynamic interplay between glial cells is just kind of another hallmark of how important these cells are for development as well as future function. And that's what we're going to talk about in my last slide. This is a depiction of a healthy brain here on the left half of the slide, and then a brain with ALS or Lou Gehrig's disease on the right portion of the slide. This is a selective loss of motor neurons that control your movement, but also eventually at the late stages of the disease, you can't do any motor output, so these individuals eventually are unable to breathe. So in the healthy brain, we've got astrocytes sampling the environment, supporting these neurons. We're going to talk about astrocytes today, sorry. Our microglia are small, and they're kind of sampling the environment as well. And then we can see what happens in the diseased brain. These microglia get activated, and they're spitting out reactive oxidative species. So we think that they're leading to the actual death of these neurons as well. But I'd really to end with this just because we've got all of these different cell types, these glial cells, supporting to have a healthy neuron here. We've got our oligodendrocyte and our myelin wraps. We've got our microglia and these other types of glia. They're all essential for the healthy function of our brains and not just in development. So today I hope I've shown you that these glia are involved not only within kind of helping them maintain brain homeostasis, but they're also really important during development, so when we're forming these synaptic connections. So thank you very much.
All right. Hello. Thanks for sticking around. So, as he said, my name is Sarah Cook. I'm not quite a PhD, but I'm getting there. I have maybe a year left. We'll see. Don't ask. And as Courtney mentioned, I actually study learning and memory, so I don't technically study the gut-brain axis, but I do hope to maybe in the future because it is kind of a very fascinating field, or I'm hoping to convince you of that today. And there's a lot that we don't know. I mean, there's a lot we don't know in general in neuroscience, but the gut brain is really this, in the past maybe 20 years, it's really becoming more popular to look into. But first, I'm actually going to play a video that's going to explain your microbiome in 3 minutes, hopefully. And it's really well made, and it's by PhD Comics, which I recommend, but hopefully I'll just let you listen. It's through this. Is this speaker connected to anything?
Microbiomes are collections of microorganisms that live together in a given environment. Every soil and sediment on Earth has a microbiome. There are microbiomes in the ocean and even in the atmosphere. In your body, there are microbiomes in your skin, in your mouth, your digestive tract, anywhere the body comes into contact with the outside world. All life on Earth evolved together, so microbes need us and we need them. We depend on microorganisms to help us break down nutrients and produce important vitamins and enzymes in our digestive system. It's estimated that there are over 1,000 different kinds of microorganisms living inside our bodies, containing about 300 times more genes than our own human cells. If you put together all the microorganisms inside you into one mass, it would weigh as much as 6 pounds. In comparison, your brain only weighs about 3 pounds. Each of us carries inside of us a unique fingerprint of microbial diversity. Your unique microbiome starts as soon as you're born and is influenced by your diet, your experiences, what you're exposed to, and where you live. In some cases, your microbiomes can even affect your mood and behavior. Scientists believe there are several pathways through which the bacteria and other microbes in your body influence your brain chemistry. For example, experiments have shown that organisms that are raised in a completely sterile environment perform differently in tasks related to learning, memory, and social behavior. Currently, scientists don't have a perfect understanding of how these microbiomes work. Although we can catalog with incredible precision and speed the number of microbes living inside of us and around us, we still have a large gap in knowing how these communities interact with each other and with their environments. To develop the kinds of tools needed to study microbial communities and how they function, we need a unified approach that draws on many different disciplines. From the inside of a single cell up to the size of our entire planet, we need new technologies and methods to help us decipher the working principles of these communities. If we can understand these complex systems, it would have a large impact on how we interact with the natural world. There are applications in agriculture and crop productivity, in climate science and carbon cycling, in sustainable energy and biofuels, and in health and medicine to treat disease or create new therapies. Understanding microbiomes may even one day help us to colonize new planets. Microbes were here on Earth before us, and they're an important part of what makes this planet habitable. If we wanted to colonize Mars, for example, it would be critical to understand the role microbes play and how they integrate with a new ecosystem. We're only just now beginning to understand the full breadth of services microbes have to offer. There's a whole invisible microbial world out there that we didn't know existed before, and we're almost at the cusp of understanding this huge part of our environment and our own bodies. Microorganism communities may be small, but they're also very important. As they say, it's the little things that make a big difference.
Right. So thank you, PhD Comics. It's a great source for fun. Okay, so I'm going to go ahead and continue with my talk. So it's estimated that only 43% of the cells in the human body are actually human. The rest is made up of our microbiome, which is, as the video said, it's bacteria, it's archaea, it's yeast, which are eukaryotes, it's fungus, it's literally everything that's not human. And as it said, our human genome roughly contains about 2 or 20,000 genes. But the genome of our microbiome is between 100 and 1,000 times more than that, which is incredible in terms of the amount of information that these bacteria are holding. And also, while our microbiome exists throughout the body, it's really the majority of microbes that are found within the intestinal lining of our gut. And there are about 1 trillion or more of them. We're not quite sure. And as the video alluded to, the composition of our gut microbiome is unique to each individual, just like our fingerprint. And while your gut microbiome might look very different to someone you're walking next to on the street, it actually looks very similar, or as similar as it can, to your mother, and that's closely followed by your siblings. And your gut today is not the same as the one that you were born with. In fact, you weren't born with much of a gut microbiome at all. Over the first 7 years of your life, you developed your microbiome, and this was impacted by how you were born, where you lived, the food you ate, and a lot of more factors, including who you interacted with, and even if you had pets or not. It's kind of cool. And as a whole, the Western world is actually losing diversity in our gut microbiome. Things like antibiotic use, spending more time indoors, and moving to cities has contributed to this loss in biodiversity. And I'll get to that a little bit later. But something that I find incredibly interesting about our microbiome is that the microbes in our gut are communicating to our brain via this gut. Oop, and I pressed the stop button. There we go. Okay. Sorry. There's a stop button on this. It's not good. The majority of neuroscience is focused along the central nervous system, which includes our brain. That's really where we know a lot about where our neurons and our glia are interacting and working together to make our bodies work. However, in the past 2 decades, many neuroscientists have come to believe that there's a second brain housed in our gut. And it even has its own nervous system, which we call the enteric nervous system, or the ENS. And this system controls our digestion. And what's cool about it is that it doesn't actually have to talk to our brain. And every other organ in our body has to talk to our brain to know how to work. But our enteric nervous system can digest and excrete regardless of what our brain tells it to do, which is kind of fascinating. And there's a reason for this. As Courtney mentioned, there's roughly 80 billion neurons in the human brain. But surprisingly, your gut actually contains 500 million neurons with at least 20 different cell types with distinct functions. Just like they were talking about with the cell types in your brain, the cells in your gut actually can contribute to different functions, which I'll get into a little bit later. But there's still so much we don't know about this. And that's why I want to study it in the future. So this communication network between the gut and the brain is termed the GutBrain Axis. And these organs are connected both physically and biochemically. And they can influence each other in a number of factors, so that our gut and what we eat can actually influence our mood and behavior, while our brain can influence our gut by regulating the nutrient delivery as well as the health of our GI tract. Let's see. And the way that they're actually able to communicate is via this vagus nerve, which is going to come at you. And this is part of our peripheral nervous system. So we're talking about 3 different nervous systems. Our central nervous system up in the brain, our enteric nervous system down in the gut, and then the peripheral nervous system, which is actually all of the nerves that are connecting your brain, your spinal cord, to your muscles and everything in your body. And you can see this red part. It actually goes from your gut to your lungs and your heart, and it goes all the way up to your brain. And it actually has a direct link to the area of your brain called the hypothalamus. And this brain region is highly associated with hunger and emotions. And so we do know that there is a direct link from our gut to our emotions. So yeah, you are what you eat. Yeah, right? We know that it sends signals in both directions, and it's currently being studied as a means of therapy for both neurological and GI problems. In particular, in one study in humans, they found that people with irritable bowel syndrome or Crohn's disease have reduced vagal tone, and that indicates that there's reduced function in this nerve in people with these diseases. And another study in mouse found that feeding them a probiotic, which I'll talk about later, but it's essentially just live bacteria, this reduced the amount of the stress hormone cortisol in their blood. And they found that if they cut the vagus nerve of these mice, the probiotic had no effect, meaning that the vagus nerve is actually really what's important for this gut-brain communication and its role in stress. And one of the most important ways that these organs communicate is using neurotransmitters that send signals in both directions. And Courtney alluded to this a bit earlier. So neurotransmitters, as we conventionally know them, are produced in the brain, and they control feelings and emotions and all sorts of things. But interestingly, many of the neurotransmitters produced in the brain are also produced by gut cells and by the trillions of microbes living there. And this includes dopamine, which Courtney talked about, which is both important in our motor system and our reward system. It also includes norepinephrine, which is important for our fight or flight response. Additionally, you may have heard of the neurotransmitter serotonin. It contributes to our feelings of happiness, and many antidepressants target serotonin specifically. But what is incredible, and what not even all neuroscientists know, that our gut bacteria actually manufacture 90 to 95% of the serotonin in your body, which is spectacular because we really think of serotonin affecting your brain. But the fact that it's coming from your gut really implies that there's a lot going on that we don't know. There's a lot to study in terms of how your gut and your brain are interacting in depression and things that are impacting our serotonin system. That's actually what I want to study down the line, but we'll see. Also, our gut microbes produce a neurotransmitter called GABA, and this is our inhibitory neurotransmitter, and it can control our fear and anxiety feelings. And studies in mice have shown that certain probiotics that can increase the production of GABA also reduce depression and anxiety-like behaviors. So there's a lot going on into serotonin and GABA research for specifically depression, but other neurological diseases as well. All right, so next up, your gut-brain axis is also connected through our immune system, like the video alluded to. Both your gut and the microbes in your gut play an important role in the inflammation, particularly by controlling what is passed into the body and what is excreted. And if your immune system is switched on for too long, this can lead to inflammation. And that can be really terrible in like autoimmune disorders and things that are kind of hyperactive, like allergies. Allergies is just your immune system fighting an antigen that you put in your body that it doesn't want. In particular, there's a toxin that is released by bacteria called liposaccharide, or LPS. And it's an inflammatory toxin. And it can cause inflammation if too much of it passes into the gut, or from the gut into the blood. And this can happen when the gut barrier is leaky. You might have heard of leaky gut syndrome. This can allow bacteria and LPS to cross over into the blood and cause inflammation. And both inflammation and high LPS has been associated with a number of brain disorders like severe depression, dementia, and schizophrenia. So this is an ongoing research field that is getting a lot of attention lately into how we can manipulate our immune system, particularly through our gut, and how that regulates other diseases and healthy brain function. Furthermore, recent studies suggest that intestinal bacteria imbalance are connected to disorders like allergies, depression, type 2 diabetes, cancer, Alzheimer's, and Parkinson's. And many of these are in part related to that inflammation I talked about and an overactive immune system. And as I mentioned, that abnormal gut function can lead to various problems with behaviors, emotion, and it can even affect your ability to sense pain and make it worse. That's more research going on to that currently, but we don't quite know how that's working. I'm actually going to focus a little bit on Parkinson's, because in the past decade, a lot of research has implicated gut health with Parkinson's. And specifically, in 2018, there was this systematic review and meta-analysis on the Parkinson's disease risk. And this found that people with IBS, or inflammatory bowel disorder or disease, are at higher risk for this neurodegenerative disorder. In fact, these digestive pathologies actually are known to precede the development of the actual neurological disorder by several years. indicating that this might actually be able to help us figure out if people have Parkinson's before they get to the point where it's obvious they have Parkinson's. And I forgot to go to the next slide, sorry. There we go. So Parkinson's disease is characterized by a buildup of this misfolded protein called alpha-synuclein in the cells of the brain. And recent studies out of Johns Hopkins have shown that, at least in mice, this alpha-synuclein protein travels from the brain to the stomach and vice versa via that vagus nerve that I talked about that's connecting the gut and the brain. And these researchers, they basically surgically cut the vagus nerve in a group, or one group of mice, and then injected the guts of these mice with this misfolded protein. And they found that mice with this severed vagus nerve showed none of the cell death side effects that were found in mice with a vagus nerve intact. And that means that this severed vagus nerve appeared to halt the misfolded protein advances into the brain, implying that we can, if we're able to regulate the actual connection between the gut and the brain, then we might be able to regulate the amount of this misfolded protein that's actually getting into the brains of people with Parkinson's. And that's going to be a long way. However, there are now therapies in clinical trials of Parkinson's patients that are using pacemakers, which people generally think about in the heart, where they make your heart beat. They're basically putting them onto this nerve and then able to regulate the frequency of the firing of this nerve and potentially treat Parkinson's and a number of other different diseases. And this is kind of starting to get popular now in just the basics of clinical research. So we'll see. So I recommend keeping an eye out for that treatment in the near future. So I've told you a lot about what happens when your gut is unhappy. So how do we keep it happy? Because that's the point of all of this. The way to do that is, unsurprisingly, to have a healthy diet. Our diet seems to be the most important influence on our gut microbiome, and a healthy diet based primarily around plants and fiber can increase the diversity within your gut microbiome, which again is a really good thing. So it's recommended to eat the rainbow. The typical Western diet, as I said, reduces our biodiversity within our microbiome, and this can actually lead to inflammation. And this diet increases the risk of diseases like obesity, type 2 diabetes, and cardiovascular disease. It's rich in processed foods that have a lot of emulsifiers and these detergents that can actually damage the intestinal lining. And this leads to leaky gut, like I talked about before. And this can also lead to systemic inflammation of your body. However, as I was talking about, the way to make your gut happy is to pump it full of good things, like prebiotics, which are essentially non-digestible dietary components that can stimulate the growth and the fermentation that other bacteria are doing in your body. So they're basically the helpers, and they serve as food for the microbiome within your gut. Furthermore, you can also eat probiotics, which are things like fermented foods, like yogurt and kombucha and kimchi and all those fun things that are sour. And these have their own source of live bacteria, and they basically contribute to the good bacteria in your gut. And then another thing that we generally associate with bacteria is antibiotics. And antibiotics are a class of drugs meant to destroy bad bacteria within and on the body. And this can be incredibly useful for killing deadly bacteria that would kill you otherwise. But it can also create problems because it's important to note that it doesn't discriminate between good and bad bacteria. It just kills it all. So if you are taking or take antibiotics, it's important to replenish your stock of good bacteria. And to do so, you eat probiotics and prebiotics and a healthy plant-rich diet and eat the rainbow. So that's all I have for you today. Cool. Oh, sorry.
Oh, microphone for me. Thanks.
Talk too fast. So, I learned so much about Glia.
Caleb, you wanna?
And I'm a neuron.
We can scooch this way, so we're directly in front of the projector light. All right, so many questions. So you in the blue first. Am I calling on people? Or are you calling on people? He's on it. All right, I won't do anything.
All right, so you talked about probiotics, and I know them as these bottles of pills at the drugstore. That's one form. Can somebody like me make any meaningful judgments on those, or is there no way to know what's good and what's useless in those?
Great question. I think the second thing that you just said is correct, and that we don't know enough about the particulars of these individual cultures that they're putting into these to know exactly what's happening. Sorry, I turned it off.
There we go.
Oh, okay. Sorry. Don't know if that makes a difference. Okay, cool. So basically, yes, they put probiotics in a pill and give it to you. However, I mean, everything that we encounter has bacteria on it. Everything that you eat. However, a lot of specific foods that are fermented are making their own bacteria, and that's what the fermentation process really is. It's bacteria eating sugars and making alcohols. So, in my opinion, I don't think there's enough research to know quite what each of those is really doing and how they're helping. I think the most important part of making your gut happy is to have a bunch of different types of bacteria. So if there's it's like a multivitamin. If there's one that has a lot of different types, that's probably going to be the most helpful. But I don't think that we know quite enough to figure out what's good for what condition.
And so much of the current research on microbiota are focused on this particular species and what this particular species, when you add it into the gut of a mouse, what does it do during a stress test? So the normal foods that we eat have way more diverse bacteria than what has actually been studied so far. So get the
So
most
I think a
you
healthy
can.
diet is more important than taking those pills.
Yeah.
But
That's
they
also
can't hurt
sometimes true about
everything.
context is important too. So sometimes
Like if you're on
I know.
antibiotics. then-
Yeah, if you're on antibiotics, or even with a totally different system, if vitamins, a lot of vitamins work best when they're in the foods that they're normally in, instead of coming in little pill form. Although pills are definitely totally helpful,
Yep.
they often make your pee more expensive, I feel like. That's my dad's favorite phrase. But so yeah, things like vitamins and probiotics, I generally just eat better.
Eat more green.
Yeah. Yeah, do you want to pass that?
A question for Dr. Dell as a developmental neuroscience. At what age in early infancy do the neurons no longer divide, and does this knowledge still hold true? And at what age does the so-called pruning process start in adolescence?
So we're actually getting a lot of information on that based on studying neurodevelopmental disorders. So I'll start with your last part about pruning. We think that's really happening, peaking in adolescence and concluding by early 20s or so. So interestingly, we think that some diseases like schizophrenia are hitting in around your 20s, because that's when pruning should conclude and perhaps continues too long, or perhaps it's not doing enough. Whereas things like autism, those would be more with when those synapses are forming, so probably initiating in your utero. And so I know that's a very broad window. Can you remind me the first part of your question?
When do neurons no longer divide in infancy?
That's still a bit of a controversy.
They actually still divide in adulthood.
Mm-hmm. So
I studied this in undergrad. I promise.
So there was a really big thought for a long time that whatever neurons you're born with, those are the neurons you have, the end. But we've found totally not true. So neurogenesis actually, which is the birthing of new neurons in your brain, actually happens throughout adulthood. It doesn't necessarily, we don't totally understand a lot of it. Your olfactory system gets a lot of new neurons because your olfactory system is right out there on the edge, so those cells are more likely to be damaged and you need new ones more often. But you have new bodies of neurons. We just don't totally understand where they go, and they can't always go to repair something after an injury like we would like them to, but it totally happens. So never, I guess, is the answer.
And the other major part where adult neurogenesis happens is a place called the dentate gyrus, and it's within your hippocampus, which is what I study, which is where learning and memory is really where we think it's consolidated. So it's kind of, it makes sense, right? Like when you're learning, you're getting more cells, and we found that like exercise and antidepressants and certain drugs and whatnot can actually increase the amount of neurogenesis happening. And so there's some studies to figure out if maybe antidepressants, the reason that they help is by regulating the neurogenesis, but we're still learning. Great question.
Thank you, all of you. I love the way you're integrating pathology and disease manifestation. And I have a question regarding glioblastoma or glioblastoma.
Yeah.
It's glia, isn't it?
Glioblastoma?
I don't know that answer.
I think
Glia.
it's
So,
Yeah.
I mean, I don't know very much about it. I just, know what I read. But if the myelin sheath is what protects, it's that gel stuff that you were saying that protects the myelin, or the sheath, is it too specific to ask you if the glioblastoma is what grows erratically around the sheath?
Well, so the glioblastoma is really scary because unlike neurons, which stop dividing more or less, I mean, they what they said is it happens on a very limited scale. So these neurons are not dividing, but glia are very, very active. And so they continue to replenish themselves. And that actually makes them kind of a dangerous population in terms of cancer. So anytime you lose your restrictions on your cell division, that's when a tumor can form. And so glioblastomas are especially scary because of that proliferative capacity. So those masses can just get very large, and that doesn't happen with the neurons very often. So that's in short why they're scarier and why they can grow so quickly. And so, but to get at your point, there's not necessarily a myelin correlate to the glioblastoma. Those glial cells are probably kind of confused and not behaving like they normally should. So the myelin component there is probably minimal, but it's more that they've kind of lost control on cell division like all of the other cancers.
And the reverse happens in multiple sclerosis when the myelin actually
You lose myelin, and there's some evidence that in MS lesions that those glial cells go in and try to repair, but they're not very good at it. And for some reasons, even in mice, upper vertebrates are not so good at clearing those things up. Fish are very good at, they can heal anything. It's just the species
Mice
difference.
too, yeah. Humans are the hardest to heal. Yeah.
I was wondering if any of you guys could tell me the difference, the difference between BDNF and NGF, what roles they play, and in particular, if there are specific diseases or problems caused by an excess.
Oh, I don't know the answer to that. So BDNF. So the question was about BDNF and NGF, what does BDNF stand for?
Brain-derived growth factor.
And then NGF is.
Somebody say it clearly.
Sorry. These are proteins.
Go ahead.
Yep.
Okay, so these are two different growth factors: brain-derived growth factor and
Sure.
nerve growth factor. I don't know.
Yeah, you've hit on something none of us study. So.
So, good question, but sorry.
Yeah.
So being a total being a total foodie, I'm really interested if you do that future study with the gut brain, like if there's a way to somehow learn or follow you if you do that, let me know.
Okay.
I was wondering, for products, when you see products for sale in the marketplace, they'll say antimicrobial. What does that really mean? Is that good? Is that bad?
It depends on what you're trying to do. Antimicrobial is just meaning that it's likely either killing or deterring microbes from being on there. So like if you're trying to make a sterile surface or something clean, so like antimicrobial soap is a good thing if you want to get bad bacteria that's going around because everyone's coughing and that's children. And like antimicrobial soap is good. Antimicrobial products if you're like cleaning things, or even I know they have cutting boards and whatnot, antimicrobial, that are made of bamboo and whatnot. But those are good things because you don't want to have that bacteria sitting there and growing and all of these things. So in terms of, to keep you healthy, it's not a bad thing. But that's within reason. I mean, not everything is going to have bacteria on it, whether you like it or not, no matter if you scrub it. It's just a matter of kind of keeping it at bay, I suppose.
Okay.
So in terms of like things that you're using a lot, like things that you're cutting, antimicrobial stuff is probably a good way to go.
Okay. You never know with those marketers.
Yeah, I mean,
marketing.
capitalism. Yeah.
I was going to ask Courtney, since we had to hold all of our questions, you had said that it's not true. We don't only use 10% of our brain. We can't use it all. It'll be a seizure.
We can't use it all at the same time.
The same time. Right. Same time. So how much are we actually? Is there a range of how much? Or
I mean, so all of your brain gets used throughout
at
your
one
life.
time. I mean,
at one time.
Yep.
I don't know that there's.
Probably depends on what
A
you're
range?
doing.
Yeah, it totally depends on what you're doing because so some activities involve more bodies of cells than other activities do. So if listening to music is one of the best ways to activate like the most different parts of your brain, as opposed to other activities, which are more purely motor or more. Basically, you're using a ton of your brain all the time. It just changes which parts of your brain you're using. And
we don't really know how much.
I don't know. It doesn't mean we don't know. I don't know. But I don't know that we've totally, completely estimated that. Yeah.
Okay, no problem. And then Caleb, I was just going to ask you why the zebra danio, I'm assuming you're referring to. I mean, goldfish, guppies, angelfish. Why is that fish the subject matter? That's great.
So the advantages are they're transparent. So I should have pointed that out. So these, until a couple of weeks, well, they start to get a little bit of pigment. But that allows, there are hundreds of zebrafish labs that study all parts of the body. And because they're transparent, we can put those labels on and track individual cells and extensions of those cells. So the transparency is huge. We're good at housing them in these elaborate aquariums.
Yeah.
They live. Yeah.
Yeah.
You can make it.
The life cycle's
fast.
fast. Yeah.
But why them specifically? I don't know. That's a good question.
Good answer.
Yeah.
Thank you.
Okay, I have the mic now. Sarah.
Yes.
When you talked about the studies where the vagus nerve was cut and various things did or didn't happen, it led me to realize that I didn't really understand what the mechanism of communication was. Is it chemical going up the vagus nerve?
Yeah.
What's going on to have this communication work or not work along that pathway?
That's a great question, and I should have made that more clear because it's electrical. It's like those tracks that both Courtney was talking about through the spinal cord, and so did Caleb. It's essentially like that, but it's not in your spinal cord, well, it kind of is, but it houses muscles in your face too, so it's not quite there. It's just a collection of all of the neurons from your gut. They send those axons, so those really long not spiny things, those really long, just like one protrusions that are sending the signals. That's what that nerve bundle is. So it's basically just all axons coming up, and those axons can go in both directions, right? So it's all electrical until it gets to the right spot, and then it turns chemical. Yeah, that's a great question.
So it's not just a single pathway. You have a whole bunch of axons making up the...
Yes, exactly.
Okay.
Yeah, it's not like that giant axon.
Yeah,
It's
so
like,
every
it's like
one of
millions of those.
Yeah, so every one of your nerves is a bundle of axons. Like that's what nerves are, is just like
That's
a
what
lot
your
of collected
spinal cord is.
axons.
Parts
So
of it.
nerves are made of neurons. It gets really confusing.
Yeah.
Cool. And I have a question for Caleb, and then I'll pass this on if you don't mind. When you were showing us the fact that the glia cells create the myelin around the
Axon, yeah.
axon, thanks. And then it speeds up the transmission along the axon. Do they know how that works? Why that works that way?
That's a brown VA.
How the myelination process happens?
No,
or?
how
Why it
it
makes it
creates the ability to have the speed enhanced, so that it being there creates the axon communicating more rapidly.
So that technical detail would have taken a little bit more setup, but it's not just one continuous piece of insulation. It's actually these little tiny sheets that are spaced at regular intervals. And
so your slide was accurate in showing the little pieces.
But in each one of those little gaps, there's groups of ion channels, and as the electricity
Does anyone know what an ion channel is? Yeah. Okay. Yeah, you're a neuroscientist, so shush. So an ion channel, every cell has a membrane, right? It has like an outside sac. And in that outside sac, there are these little pores, or like little doors, and they open and they close, and they're called ion channels, and they let through ions. So like ions like sodium or calcium or any ion. Exactly. So when we're talking about a neuron actually firing or sending an electrical signal, it's actually propagating by the opening and closing of those channels and the changing of all
the electrical
the electrical balance between the inside and the outside of the cell. Does that sort of make sense to everybody?
Because some of those ions are positive, some are negative, and the way that they go can literally make that signal go or stop it completely. Yeah.
So those windows are opening it in succession as the electricity flows down that axon. So each one of those little gaps, it fires
super efficient gaps. Yeah.
Well, and so I have if I can use a metaphor real quick, because I love this metaphor. So picture you have a tube, like a really big hose, and that's your axon. And there are a lot of holes in the axon. Now, if you put water through that hose and all the holes are just open, it'll just like be like a slow sprinkler, right? But if you take the same amount of water and you close some of those holes, it'll just come out the few ones and go much faster down the...
Self-propelling.
Yep.
Are we doing any better at being able to repair broken neurons, like the spinal column?
Yes. I mean, always yes. Because a lot of people are working on it. Yeah. So the question was whether we're getting better at repairing neurons and spinal cords and stuff. So yeah, there are a lot of people working on that and figuring things out. It's just a really hard problem because not only do you need to, if you're dealing with a neurodegenerative disorder where you're actually losing neurons, so neurons are dying, you have to find a way to sort of replace the function of those neurons, and if that's using new neurons that your brain is using, you have to make sure that neuron gets to the right place, communicates to the right cells, talks in the right way. Like it's very complex. So yes, slowly.
I think the supportive environment maybe is more important. It's really tough to replace those neurons, but if we can make those neurons happier by putting healthy glia, by having a more, sustainable diet, these kind of things, we have to keep those neurons alive is the first step, I think, in curbing these kind of
Keeping neurons alive is way easier than bringing neurons back to life. Yeah.
We are getting good at manipulating our spinal column, though. Yeah. And so, taking away the brain part of it all, we can essentially make things move just by stimulating different nerves within the spinal column, and that's really interesting because we're making people able to walk that are paralyzed from their legs down, and that's starting to become a big deal. But it's still not to the point where we're curing anything, right? We're just putting another tool in to make it work. But
and the other sort of natural advantage is that brains are like I said, are changing all the time, right? So. They're super plastic, and plastic not in the sense that they'll go later on to poison a poor sea turtle, but they're changing all the time, right? So you can have parts of your brain co-opted to a new process or something like that. And that there's some really cool, if you get in a YouTube hole about neuroprosthetic adaptations, basically with brain function, we can sort of relearn how to drive, say, mechanical arm or something. There was a cool video I watched about a woman who was paralyzed, but they put all these electrodes in this part of her brain that does movement. And over time, she figured out, sort of just by thinking, how to move this arm that they attached her to, essentially. So that's really early stuff, but it's totally possible in the future.
I was wondering for any of you if you could speak at all about what happens neurologically when, say, someone experiences trauma from the outside, like child abuse.
Yeah, that's, sucks. Firstly, no one should ever have to go through that. But basically, it has to do with the learning and with really, really emotional moments, like emotional memories. We learn those really well and right away, and they stay there. So if you have a really emotional memory that's associated with, you know, these 8 sensory cues, like fireworks for a lot of people who have gone overseas and have PTSD from serving, or, you know, with child abuse, if there was like a fire involved or something, fire might be a trigger for you in the future because those things are so well attached, because when your brain learned it, it also had the motivation, because it was an emotional event, to really, really stamp it in there.
Right. For survival and for certain things. Things that, yeah.
So yeah, if something's really dangerous to you in your childhood, you might survive better if you avoid that stuff in the future. But it doesn't just take in the salient features, it takes in all of the features of that experience, right? So if there was, like, I don't know, a Pez container around you when something really bad happened when you were a kid, maybe Pez containers are bad. That doesn't mean that Pez containers caused your trauma, it just means it was there and it's in the associated context, right? So.
And sometimes a lot of the trauma that people are dealing with decades later can have to do with the fact that that memory hasn't actually been consolidated or been able to be, because it's so strong. And sometimes that can make people block out the memory. But there's a lot of research going into like how we can look at memory and try to use these memories but properly consolidate them so that you're able to move past them, essentially. And there's a few cognitive behavioral therapies too that are looking into that, and we don't even know how they work, but they seem to. Yeah, one of them is called EMDR, iRapid, yeah, and it's awesome. I use it and I think it's phenomenal, but we don't know how that works. But we think it has to do with the fact that with EMDR, it's eye movement rapid. I don't know. Basically, you make your eyes go from back and forth, and you can do it with these paddles that vibrate or sounds. And when you close your eyes just go back and forth. And they found that actually somehow, we don't know how, is consolidating your memory. So it's taking these short-term memories or longer-term memories and really allowing your brain to process them and move past them. And it seems to work, or it's helping me. Yeah, it's pretty cool, but there's some research.
Pseudoscience. There's some people say that it's woo-woo.
EMDR?
Yes.
It's possible. I mean, everything works for certain people. Yeah, and I mean, cognitive behavioral therapy too. It really depends on the person and who you're working with too. At least with me, I really like my therapist, and I feel very supported with her. So it's kind of like it's partly the environment, but I've been able to process things. But it does. I mean, we don't know what's happening at the molecular level, and as a molecular biologist, that's what I'm curious about. But that's how we learn, right? We observe something, we see something's different, and then we try to figure out why. And maybe it works, maybe it doesn't. But, yeah.
Cognitive behavioral therapy is accepted and apparently works. But never mind.
That's okay.
Okay, so I have one question for Caleb, but one for all three of you. In terms of like glial cells, you talked about their role in terms of learning, making the signals go faster. Are there other mechanisms? Because I thought I saw one photo where it was like a glial cell connecting two axons together or something. Are there more mechanisms where glial cells help with learning, so that it's not just synapses and neurons, it's glial cells also?
Yes.
So I originally in this talk was going to talk about how microglia can actually help shape synaptic connections as well. So instead of just like consuming whole cells or debris or things like that, and I showed you how that microglia is just kind of just surveilling its environment. So there's some fascinating new data that somehow that microglia is sensing well, I showed you with the myelin sheath, but also with these axons and different aspects of the neuron. It knows which little things, those little nubbins to prune off. And somehow it's got the knowledge that these nubbins are correct and these ones have to go. And so that implicates the glial cells in a deeper way in how the neuroplasticity is taking place. Does that make sense? So it's not just interacting with other glia, it's also with the neurons themselves. So yeah, there's some fascinating stuff going on.
And one for all three of you. If you guys have heard of mini brains, people are working on, how excited are you about it, or how do you?
You want to describe mini brains? I don't actually know how to
I
describe
mean,
it.
or just excited by, so mini brains are just lab-grown small brains.
Yeah, so there's organelles that they're starting to grow that have connections, and they're able to see that one cell is talking to another cell. They're not quite brains yet, but they're kind of like, we're getting there. Like, we're growing organs on dishes, and this is the first step to getting to a brain. It's exciting, but there's limitations, obviously. The thing that I'm really excited about is brain on a chip, and that we're using computational biology and like cellular and molecular biology to really bridge those gaps to make it so that we don't have to kill as many mice to look at these cells and like play with the circuits. We can just have it on a chip and use different drugs and use different like disease mechanisms and put it into this chip. That's going to be a while, but that's what I'm looking forward to, because then that takes out everything. I mean, that takes out the whole, I don't know, process of killing animals.
Earlier, we mentioned multiple sclerosis and other diseases where the myelin disappears. Why does the neuron eventually die, like in multiple sclerosis, when there are other nerve cells that do not have at all myelin and they don't die? Do we know?
I didn't cover that in enough detail. So the myelin is not just an electrical insulator. It's providing nutrients and growth signals and actual physical protection, like having a Band-Aid around. So it is interesting that a lot of neurons are not myelinated, and you're right, they do just fine. It may be that having that myelin protection for such a long time and the removal makes it especially vulnerable. So, but I would probably suggest that the loss of all those nutrients and the kind of just being blanketed in that protection is probably why they specifically lose.
You know, so much of what you're talking about has been the transmitting from an external organ up to the brain where it's processed. Anybody studying what the processing is?
A lot of people do. Yeah. Yes, I mean, you're working in a central, I'm working in the periphery because I'm trying to figure out how taste buds are talking to the central nervous system, but they're definitely, I mean, the periphery. But yeah, tons of people are working in the brain.
Right. And we study just cells within the brains themselves, not taking out these brain regions and looking at those cells in particular and trying to apply what that means. There's some, there's some caveats involved with that. But there's so much we don't know about how things are processed that is really the point of it all, right? Like with memories, we know that like there's different parts of the brain associated with memories. I mean, your visual system, obviously, your auditory system, even your olfactory system, what you're smelling. But like all those memories have to be consolidated somewhere, right? And in short term versus long term, contextual versus declarative, like there's so many different ways that has to come into play. And all of the timing and the spatial components and the resolution of that's what we're really focused on. Because, I mean, the brain is, otherwise it's just a bunch of cells talking to each other, right? Like I don't necessarily study circuits. That's how the brain works, right? One part talks to the other. I study within the cell itself how it's regulated. But even there, we don't know much. I mean, there's so much that we don't know at every level that it's kind of hard to extrapolate. But we're trying.
But yeah, there are tons of people working in the brain. I prefer the periphery myself, but
sensory stuff's cool, but.
We've touched on how changing our environment consciously, changing our sensory inputs, changing our cognitive behaviors can impact the neurons, either through the gut or through direct interaction. The glia, are they kind of just working to their own agenda, or is there a way that we can externally influence their activity?
Great question.
That's an awesome question.
I don't even know how to answer that. I don't. They're still their end goal is still going back to the neurons, and so that's how quickly I can sum that up. I mean, I don't know. Let's see here. We do know from some studies that like learning and memory. So they inhibited a mouse's ability to make new myelin, and those mice were not able to make new memories. So we know that myelin is essential for those new formations. And so we know that it's active, I guess, but I don't know how to attack how we can influence our glia. But I'm going to think about that a lot.
That's great, yeah.
I mean, we probably can.
Yeah.
But it probably winds down to the same things that keep all your other cells healthy and happy. So, yeah.
You mentioned that squid don't have myelin on their axes. How far back in the evolutionary tree does the trick of myelinization go?
It's vertebrates, but not all vertebrates, I believe. So I don't know the exact branch here. So interestingly, there are some glia in such things like people study C. elegans. It's a type of nematode, a worm, and they have glia that are similar to oligodendrocytes. So they're ensheathing glia, so they'll wrap an axon just like an oligodendrocyte will. So even though it looks like the vertebrate branch is where this all evolved, even invertebrates have glia and glia that are similar in function to oligodendrocytes. So it's an ancient adaptation to nervous system.
Is it easy to describe the different characteristics or outcomes that we associate with different neurotransmitters? Is that different action potential or different proportions or different neurons, or how do we associate certain characteristics with certain neurotransmitters? And also, are we able to categorize them in different combinations and proportions of neurotransmitters?
This is a complicated question. I mean, in short, so neurons are diverse in that they use a lot of different neurotransmitters, but each neuron uses its own sort of like fingerprint of neurotransmitters. So some just use dopamine, some use like dopamine and something else, some use serotonin and something else. Like it just depends what that neurotransmitter is doing. Like the neurotransmitter is just a chemical, right? It's not doing any, it's not magic on its own. But it contextually, if it's coming from a neuron that uses it to a neuron that uses it, that's going to be significant. But outside of the circuits where it's involved, it doesn't have any inherent meaning or purpose. Does that sort of answer your question?
And there's in particular brain regions, there's subtypes of neurons or populations of neurons that are like dopamine neurons. So they signal to dopamine neurons too, and they talk via dopamine. But they might be talking to a neuron with dopamine, but that neuron itself might have GABA. So it might, they're all kind of connected. And as I was saying, the really important aspects is the temporal and there's the spatial aspects of these. So when it fires and where it's firing. And that's what we can kind of either control using chemicals or electrical manipulations, like when we're experimenting, or using drugs and whatnot. So there's drugs that can regulate specific neurotransmitters themselves, the amounts that are there, the amounts that are readily available. So like selective serotonin reuptake inhibitors, like SSRIs. Those, they don't directly act on serotonin, but their purpose is to make more serotonin available. But it has to be in the specific place for that to even work for depression.
And how do you get granularity on associating characteristics with the different neurotransmitters?
In terms of activity or behavior?
Any way you want to go about it?
Like what is serotonin related to specifically?
Well, we have a presumption of the function of serotonin, and as you say, it's a chemical used in different parts of the body, a function of dopamine, a function, and I've been really surprised to see that ketamine now works on glutamate, which I thought was the most boring of all.
No, that's what I study glutamate. We're now learning that
it's boring.
It's
cool.
Exciting, actually. It's the excitatory neurotransmitter. Sorry.
That was nerdy. That was pretty nerdy.
Pretty bad. So I heard that. Yeah.
Characteristics from the different neurotransmitters.
I guess I'm wondering which characteristics, like what systems it influences.
No, dopamine we associate with focus,
movement,
with motivation, with anticipation.
Right, and serotonin we associate with mood.
Affect, norepinephrine has its case of. How do we derive?
That's all based on what circuit it's a part of, right? So the basis of how your brain is functioning for everything is a circuit, and so you can say like acetylcholine is another neurotransmitter, and the cholinergic system you can figure out. Oh, this cell has acetylcholine. This one has acetylcholine. This one has acetylcholine. So you look at all of those and follow them and then ask what are they up to. And sometimes they do the same things and sometimes they don't. So it's all based on what the purpose of the circuit is and what tools it uses. For example, serotonin in humans is associated with mood and depression. So we use tools that make it more available, like Sarah said, to ease depressive symptoms, right? But in invertebrates, I have a friend who studies stalk-eyed flies. So they're these little flies that have these really big eye stalks, and they fight all the time. And they figure out, they usually measure their eye stalks against each other, and whoever has the bigger eye stalks gets to win the fight. Very boring. But basically, he did a lot of experiments changing the serotonin in their system, and in their system, an increase of serotonin actually makes them more aggressive. So it can have totally different, the same drug, the same thing can have totally different effects on a totally different system, right? So it's all based on what that circuit does.
Even within the neurotransmitter itself, within the human, we were talking about dopamine cells in Parkinson's and how that's affecting motor output. But then we also know that dopamine's involved with regulating our reward behavior and addiction and all of these things. But there are different pathways that are involved in different parts of the brain.
Right.
So it's the chemical itself is the tool, but then where and when it's being used is really what's going to regulate what the output is.
Or serotonin has all the associations we have, but it's also the neurotransmitter that your sour sensing cells in your taste bud use to communicate sour, the presence of sour. Yeah. So that's not related at all, And in your skin cells, there are certain skin cells that communicate to nerve fibers that say, "Oh, I'm being touched right now," and they also use serotonin. So it's
an MSG.
Yeah, exactly. Or there's cells that use ATP, which is usually you think of as the energy source for the cell, but it can also be used as a neurotransmitter. And all of taste signaling depends on that. But ATP signaling is also important for a lot of pain regulation. So there have actually been pain drugs that have been developed to prevent ATP signaling, but people don't take them because it also messes with taste. So there's nothing inherent about that. Like Sarah said, it's the tool, and it just depends in which neuron's proverbial hands it's in.
Mm-hmm. And drugs targeting these can actually, like she was saying, can really affect these systems that they're not trying to affect.
Yeah. Because all of our drugs are a big brush where we want to be really detailed, but we can't.
Yeah.
So.
We're trying.
Maybe 2 more questions and then we'll call it.
Okay, I just want to answer one of the previous questions, that why would we use Denio instead of any other species of fish? I work with zebrafish too. Hey guys. We're all colleagues in the same building. So one of the major reason is because we share about 70% of our genome with them. So they're the closest probably to all the species of fish. And there is another one, but that one is closer, but it's very difficult to grow it in the lab and to get embryos. So that's probably why it's not that popular of a model. But the zebrafish is, 70% of genome shared with us, with humans, and then about 85% of that 70% are genes that are linked to diseases in humans. So that's why it's so easy to study genetic diseases in zebrafish. And also we have the numbers. We can get up to 500 embryos from one single pair. So we can have a lot of material to study and to different experiments.
Hello. I've had 2 brain surgeries for epilepsy and wanted to share an experience and ask a question. This was really confusing to everybody on staff, all the neuro staff and everybody. During my last surgery, at some point, I got really dizzy. My vision was very blurry, and it just wasn't going away at all. Everybody was confused. My eye exams were completely normal. And finally, speaking with my neurologist, I think he sorted it out. He was telling me, I think this is all going to take care of itself because after he gave me he's like, I like to give people this creepy analogy. He said, I'm processing a bunch of new information. He said, when you go outside of an elevator and you see a carpet, you don't remember what color it is. And so if you go outside of an elevator and you see a bloody body, you remember everything about it. And so he was saying everything was new information. So I was like, this wall isn't new information. And he gave me that analogy. So basically a wall, street lights, sidewalks, and all that were new information. And yeah, so that confused everybody. And I think that's probably the best answer. I think that was actually right. He was right on with that, because I mean, I've shut down downtown. To do something fun for the first time after my last surgery, I walked into a tattered cover, and it was so overwhelming I had to walk out.
Yeah.
And but my question was, I was told that, because I got the second surgery out of concerns with aging with epilepsy, and I was told that I would have less chance of going into dementia by having this surgery than I would if I haven't. Do you have any idea why that is?
I mean, the short answer is no.
Okay.
The problems you were having with the vision and the, that's fascinating. I study learning memory, and it sounds like a visual memory problem where you forgot what you saw, or your brain forgot what you saw in some context, right? But in the visual aspect itself. So it's like when you were seeing these new things, it's like you didn't have the other things to use as context, right?
My
And so it
entire
was
right temporal
overwhelming.
lobe, I don't have right temporal lobe
Yeah.
anymore, so.
And so I imagine it affected somehow your visual memory.
Yeah.
And then it maybe took time for you to be sensitized to this reaction, I imagine. Like, did it ever actually go away, or did you just get better at it?
Yes, it did. I was very scared.
That's
I wasn't
good. Yeah, I
able to
imagine.
go back
Yeah.
to work.
Yeah, because everything
but
being new and
I thought
terrifying.
I'd share that with you just
Yeah.
because
That's
I'm
interesting,
sure
yeah.
there's other people around the country that are like, what's going on?
Yeah,
What do we tell this patient?
I imagine.
So,
But I'm glad that it went away. But
Well,
yeah.
and in addition to
Jeez.
doing things like processing what you're seeing in front of you, there are whole sections of your brain figuring out this part is important, this part is not.
Yeah.
So the part of your brain which is really distributed across your brain. It's not. I don't know of a particular locus that really controls that, but that may be just something that I don't know, not something that we as humans don't know. But if you're perturbing your attention system, the system that says, hey, pay attention to these 5 things, but don't pay attention to what you ate for breakfast because it doesn't matter. That may have been perturbed too, and if that is perturbed, then you could, might be, I don't know what of this information that I'm taking in is actually salient, which is also something that goes wrong in, say, an autistic brain, is because part of the thought there is that they're not pruning enough, so they have too many connections between neurons, and the thought is, there's no way to tell, this is salient information, this is not important to me right now. So if all of it's coming in and you're processing it with the same attention, that was really hard.
Yeah, I imagine.
I believe that. Yeah.
Yeah.
Autistic people their whole life, right? That overwhelmingness.
Yeah.
No, there's a lot of good therapies now where instead of
we're getting better at it.
Instead of sheltering a child from experience, that it's actually exposure to these things can help
kind of
things
sensitize,
better.
yeah.
So that's that's certainly
Maybe before, but now.
yeah.
Well, and autism is also one of those things that it's a big ol' umbrella of totally, so it's symptom defined,
Right. So
not
it might be
necessarily.
different things happening in the brain. Yeah.
Right, exactly. So there's something that's going sort of off, if you want to say that, but I don't know. Normal is a kind of washing machine setting, not a person. But like the things that are different about autistic folks might be different in this autistic person from that autistic person. The thing that caused the difference might be totally, completely different. It's just causing similar symptoms. So
It's incredibly hard to study.
Yeah.
It's hard to really focus on what you want to manipulate to make it better if everyone has different things. Yeah.
So my brain was completely lit up by this talk. Thank you all, and let's show our appreciation to our speakers.
thank you guys.